system is used every day in our body. It's called the immune system.
Okay.
And why this is important is, imagine you're an immune cell, and you're just came out of a breeding place where all these immune cells are replicating, and you're like a newborn cell, and you come out and you have to go get an experience of the environment. And your job is to go look in the environment and find something that doesn't belong in the environment, and then make an antibody against it. That's what the immune cell does, okay?
Right.
So the idea is, I go out and let's say there's this virus that's never been there before, and I can recognize it in some way. Then what happens is, over a 3-day period, my interaction with that virus results in the development of a new gene that makes the antibody that matches that virus. So what it says is our immune system is an example of using this mechanism of evolution, and that is, cells are intelligent. They go out and read the environment, and they learn from that environment through a process of creating new genes. And much more critically, what we've recognized now as of late, and this is very important new stuff in the sense it's not new, it's 11 years old, but it hasn't even entered the mainstream thought yet, is really a work by a British geneticist by the name of John Cairns, and I think it's one of the most important biological papers in 100 years for the reason.
I wanted to explore Cairns' work closely, because that's that very interesting lactose bacteria experiment, isn't it?
Yes.
Tell us about Cairns' work.
Cairns' paper was called The Origin of Mutants, and it was published in Nature, so that's like a fundamental biomedical journal. And what he showed was that organisms under a stressful situation can have a select group of genes related to the stress, alter the genetic, mutate them. In other words, mutate genes that are related to the stress that the organism has experienced, and the result of it ends up with a gene modification that deals with the stress. So basically, an organism encounters stress and then modifies its genes to accommodate the stress.
Right.
And it's not an accident. It's not a random event, because when they encounter the stress, they don't mutate all of their genes. They tend to mutate very specific groups of genes that are directly related to the stress. And why this is important is now it's not a random mutation. It's called an adaptive mutation. And adaptive mutations take the randomness out of life, and it says that organisms are, by their nature, always complementing the environment that they find themselves in.
Wow, so it's intelligent mutation.
Absolutely.
It's directed
we're getting
mutation.
to a knowledge that, and there's all these mechanisms that have just come about, an understanding of these mechanisms of things in the nucleus, program devices that allow for rewriting of the genes, an active rewriting process that allows new genes to evolve, while not even affecting our working genes. I mean, if we have genes that give us life, if we alter one of those important genes, and the alteration is like a negative alteration, then you immediately die. What we now find out is that there are like extra genes that we can use as play to try out new things, and if they work, then they can supplant or replace the existing one and improve the system. And these are all new writing mechanisms. So they classify these new genes. They have the name for this new class of genes. It's called genetic engineering genes. Genes that are responsible for rewriting the program and to keep the organisms always adapting to the environment.
That would explain how punctuated equilibrium then could happen, where they find through the fossil record that an environmental shift or climatic shift happens or what have you, and then there's a whole bunch of new adaptations, and then they level off, and then the next catastrophe happens, and there's a whole bunch of new adaptations because the organisms are learning to adapt to their new conditions in their environment, right? And they can do
very
so
rapidly,
in a directed way.
Way faster than Darwinian biology can allow. So this brings in this other factors that are directing, influencing the outcome of the product here.
More on these implications, some of the new findings in biologists with biologist Bruce Lipton. I'm Laura Lee. We get back to biologist Bruce Lipton right here on the Laura Lee Show. And that call from Ernst, finally. We found you. Hi, Ernst.
Hey, what a wonderful show you have. I'm listening to it off of KFRE.
Oh, out of Fresno.
Yes. I'm a first-time listener and first-time caller. I also consider myself an average individual, but I was curious, is it possible to take a moment to explain what kind of a code is in DNA? I understand that there's a spiral and there's bars. What is it? Is it like some kind of molecule?
Do you want me to try to answer that?
Oh, yes, please.
Oh, yeah, okay. Well, I was talking about poppet beads that kids make necklaces out of, where they pop the beads one into the other and they link them together like a chain.
Right.
Okay. Imagine a chain of poppet beads where the beads are amino acids. These are the, when you go to the health food store and you get things like glycine or proline and things like that to, beef up your diet with different amino acids.
Right.
The beaded chains are proteins, and the individual beads are amino acids. So basically, proteins are long beaded chains of amino acids. There are 100,000 different proteins. The difference between the proteins are two things: how many beads are in the chain, and then more critically, the sequence of the beads down the length of the chain. So I have 20 different colored beads, 20 different amino acids, and I can arrange them in different length chains and in different sequences of colors, okay? Mm-hmm. And these proteins give up the body its shape and all of its characteristics. Basically, it says you're made out of building blocks called proteins, and the proteins are beaded chains, and the difference between the proteins are the sequence of the beads and the length.
Bruce, is it like that, just to draw an analogy, is it like words building sentences and building paragraphs, because you've got, however many amino acids? Yeah, those 4 amino acids, and then the combination of them create words, and then you string so many words together, and you get sentences. You string so many sentences together, you get paragraphs. You get meaning out of that.
Yeah, well, basically, that's putting it in the verbal.
I'm an English lit major, so those are my analogies that I have to draw from.
So it's just stringing units together, but then the words have to come in the right sequence, or the sentence doesn't make sense.
Or it's meaningless, yeah.
And the words are the amino acids, so that you have to string them together in the right sequence. But the issue about proteins is that proteins, like parts on the car, wear out. And if they wear out, you got to replace them, and you got to replace them with the exact same part. So all of a sudden, now we have a requirement of memory of how do I know which amino acids go, what's the sequence? I don't have the memory of that. So this is why DNA becomes important, because DNA is a very stable molecule. It's so stable that they can isolate DNA from fossils 50,000 years old and use them in molecular assays and make proteins from the DNA blueprint from an organism that died 50,000 years ago.
What makes DNA so stable? What is it special?
It's just its molecular structure. It's like he was talking about. It looks like a ladder twisted in a helix with little crossbars that hold the ladder together. Okay, so basically it's a very stable molecule, so it can last thousands to 100,000 years.
This is good, or who knows what the next generation would look like, right? You could mix and match at will.
So the bottom line is now that we know that the rungs of the ladder represent a code of the amino acids. So the rungs of the ladder describe which amino acid comes after which amino acid comes after which amino acid. So the bottom line is this: the DNA is a very stable molecule that has a blueprint of the sequences of amino acids for each individual protein, and that each one of those sequences is called a gene. So a gene represents the sequence of the code for the amino acids for one protein. That's a gene.
Now, these designer genes, these genes that are able to go in and rewrite the code at will or when necessary, what makes them able to do that?
Well, again, it's a response to the environment. Stressful situations result in an activation, like the environmental signal that says stress, like an adrenaline rush in your body. Okay, activates the genes in that system for rewriting, and then somehow or other, which is not really clear, direct them to work on certain functions, not just to overhaul the whole system, but to look for like an error or glitch in the system that it can overcome to see if it can overcome the stress. So it's not random. It's all of a sudden, it's not just writing new genes, but it's selectively writing which group of genes are going to be affected by it.
Where does that intelligence come from? How is it expressed? How are those genes built? If a gene is simply a matter of this string of proteins,
yeah.
what's
No,
special about
the gene
it?
isn't the string of proteins. The gene is a DNA molecule, and that's different than protein. DNA and protein.
Well, I thought that the amino acids and then blah, blah, blah.
So, okay, yeah.
What is, you break it down to its basic parts, but what is special about these these genes that are able to go in and rewrite the program? How can they do that?
Well, this is the newest stuff that they're looking at right now. Mechanisms of how like they unplug genes at certain, they can unplug the DNA at certain place and replug it in at another place, and the disconnections and the reconnections are programmed in some way. And this is all leading edge stuff right now because it's only been in the last couple of years that they actually formally recognize this class of genes for their function.
Are these the jumping genes, the bacteria that are able to?
There's part of it. Some of them are called transposons. Some of them are. Yeah, the whole era understanding of DNA stuff has gone very, very different as of late because we now recognize that organisms pick up DNA from the environment and test it out.
Right. Now, the classic case that I've heard about for some years is that you have a set of bacteria that have acquired immunity to a certain bactericide, for example, or to a certain...
they learn that.
Right. So they become immune to all the poisons that we're throwing at them. So it needs new bacteria that don't have this immunity, haven't acquired it yet. And these bacteria get together and they can actually trade genes. They can actually make a copy of the gene that gives it the immunity to the poison and give it to the next one. And jumping genes. And who was the woman scientist who discovered this? Barbara McClintock. And what was her claim to fame? And she discovered this, and for years they ignored her. They said,
ignored her.
You can't be.
She identified what are now called transposons. She called them that, okay? And now people ignored them because, again, it meant that there was an intelligence to the system, and they were locked in the Darwinian point of view, which says there was no intelligence, just random accidents. So when she came up with intelligence, they looked at her and said, well, not only is that a stupid idea, but you're a woman scientist and we won't listen to you anyway. Ultimately, though, she won out. She got a Nobel Prize for it, but it was many years of being put down.
How many years did she go through that?
I don't know, 20, 30 years, I guess. I'm not really sure.
So look at how we, because of having the blinders on, how we missed 20 or 30 years of progress and of getting that new paradigm thinking, back into the
right
saddle.
Now. That was her work is 40 or more, 50 years ago at least. And we're just talking about it now again. It's just coming up as current. The current new rage is at work right now. And ultimately what this is going to really lead to is that the organism, the behavior and the expression of an organism is really the result of a dynamic interaction between the organism and the environment that the organism lives in, and so that organisms reflect the environment. Why this is critical is, with this knowledge, it says, then you must really take care of the environment you live in, because if you alter this environment, you ultimately will alter biology and human biology so that we'll become different then.
And going for the so-called designer genes where we want our children to be this, that, and the other, we're going about it exactly the wrong way.
Absolutely.
Because it's not the gene. The gene would have many possibilities. Which possibility do you want to draw out of the hat? Well, it depends on how your environment is stimulating that gene, isn't it? Let's take a break and let's come back with, we have questions for you from our callers at 800-485-1922 for biologist Bruce Lipton. I'm Lily. We continue with biologist Bruce Lipton. Important stuff, I think, is being put on the table, and the implications of how we conduct our world, what we do with our environment, how we grow the future are immense. Tom in State College. What state would that be, Tom? And welcome to the show.
Pardon?
What city are you in?
State College, Pennsylvania.
Oh, welcome.
All right, thanks. Yeah, thanks for having me on.
You bet. Go ahead.
This is something I'm interested in. I could talk for hours myself, but I'll try and be brief and make some sense.
Take your time. We've got time. Go ahead.
Well, I agree with that maybe mainstream science puts a lot of emphasis on genes, but it seems to still be necessary to interpret.
Yeah, it is really a work by a British geneticist by the name of John Cairns, and I think it's one of the most important biological papers in 100 years for the reason.
I wanted to explore Cairns' work closely, because that's that very interesting lactose bacteria experiment, isn't it?
Yes.
Tell us about Cairns' work.
Well, Cairns' paper, that virus results in the development of a new gene that makes the antibody that matches that virus. So what it says is our immune system is an example of using this mechanism of evolution, and that is cells are intelligent. They go out and read the environment, and they learn from that environment through a process of creating new genes. And much more critically, what we've recognized now as of late, and this is very important new stuff in the sense it's not new, it's 11 years old, but it hasn't even entered the mainstream thought, is to go look in the environment and find something that doesn't belong in the environment and then make an antibody against it. That's what the immune cell does, okay?
Right.
So the idea is I go out and let's say there's this virus that's never been there before and I can recognize it in some way, then what happens is over a 3-day period, my interaction with them is used every day in our body. It's called the immune system.
Okay.
And why this is important is imagine you're an immune cell and you're just came out of a breeding place where all these immune cells are replicating, and you're like a newborn cell, and you come out and you have to go get an experience of the environment. Your job